We we have a pretty low barrier to entry. When we talk with other scientists and learn about the problems that they have in their area of expertise, uh, we tend to be maybe naive is the word, or maybe just courageous about entering into new areas. So I was trained as a natural product chemist. I'm really a chemist at heart, but my lab now, like I said, engineers organisms all across the tree of life. A lot of those projects came from an initial invention that we had where we learned to engineer a speciation event. So we could take an organism, any sexually reproducing organism, and genetically engineer it, not to sterilize it, but to make it so that it could only reproduce with similarly engineered organisms. But if it ever tried to mate with the wild version, none of the offspring would survive. So this has important implications in, for example, the containment of transgenes in GMO crops, but it also has applications in genetic pest control. Because if we do this to a mosquito and release the male mosquitoes, any female that they mate with out in the wild won't have offspring. And I think I don't know if you want me to bring up this because you know about it, but an unexpected result from our lab, we were trying to accelerate the engineering of large fish like common carp. And we are experimenting with an approach called surrogate host transplantations, where you take the stem cells that make the sperm and eggs from one species and you put those into a different species. And we wanted to have minnows that would basically give birth to carp because that would allow us to engineer the carp genome in a much faster timeline. Minnows mature to sexual reproduction in only 3 months, whereas carp take 2 years. In the course of that research, we had a negative control fail, and we learned that we could fertilize carp eggs with minnow sperm. So we made a new type of fish. We called it a marp. It's a new type of hybrid. It's kind of like when you mix a donkey and a horse and you get a mule. And these marps appear to be sterile, and they could actually be useful for doing things like pollution control out in the environment because they would mitigate the risk of spreading transgenes since they're completely sterile.